Welcome, everyone. I'm Ryan Bernhardt. I'm the CEO of Biosero, and I want to thank you very much for taking the time to join us today, where we're going to talk about how Biosero is automating science to accelerate innovation and unlock the potential for science in your laboratory. Historically, science has been notoriously challenging to automate. Each research group or lab has different needs, different applications, different space and budget constraints, different levels of expertise. And even when the research goals are similar, the choices vary. The choice of instrument, the type of technique that would be used in the process, and the science keeps evolving. So once automation has been installed, it needs to be able to evolve methods and configurations. And for many labs, the cost of automation is still very high, not only in the sense of dollars, but also in the sense of the resources and expertise that's needed, as well as the time it takes to implement and validate the automation solution. And finally, automation in the laboratory historically has been seen as complex, where scientists find it many times intimidating to use or even hard to find training. But lab automation has the promise to deliver more with less. By extending the hours of productivity for scientists within the laboratory, with the use of computational tools, it provides better shots on goal. It allows for rapid iteration, and with this, we can better predict the future because we have access to real-time insights as that data is coming off. With automation, we have better data integrity, and we are able to overcome some of the needs that came out of the pandemic, where we're faced with the ability to run more samples with less people available in the laboratory while still being able to visualize the results and the progress as it's progressing. And finally, automation allows for enhanced data security, as well as the seamless ability to replicate that data, which, as many of you know, the replication or duplication of data in experimental results is something that's very, very challenging to do. The results of automation have never been more important. For many of the world's largest life science organizations, the mission is to accelerate getting innovation and drugs to the market faster and more cost-effective than ever before. Many laboratories have goals of reducing that cost by 50% while also reducing the time to get there by 50% as well. In this one case study from 2024 from a large pharma customer that is implementing integrated automation that's powered by Green Button Go, gave us some statistics that this automation has allowed them to reduce their downstream process and assay development time by 75%. It's increased the existing equipment capacity in their laboratories by over 400% by enabling parallel processing and variable-driven robotic process control. And it's also maximized the productivity of their scientific staff by over 200%, freeing them up to focus on other things. The rewards of automation are great, but there is a process to get there. The days of scientists standing in front of a standalone instrument or doing manual tasks to really move the needle forward are over. Today, it is very much hinges on the integration of robotic arms, instrumentation, connected into software and data tools, and leveraging AI and machine learning as part of that integrated process. Today, lab automation has now become its own specialization inside of our scientific operations. In fact, it's hard to imagine a future where science is performed in the absence of automation as we move forward. And just as the technology and the integration capabilities have evolved over the last several years, Biosero has also evolved as a partner in enabling and supporting lab automation. Biosero was founded in 2003, and from 2003 to 2013, Biosero was very much distributing devices that could be used in a semi-automated fashion within the laboratory. And in 2013, Biosero launched our Green Button Go software, and it has been our mission since to focus on providing and enabling integrated scientific solutions that are powered by Green Button Go. Today, we have over 1,000 laboratories that are leveraging Green Button Go. We have more than 3,000 installations of Green Button Go software, and I'm proud to say we're trusted by all 20 of the top 20 pharma companies around the world who are leveraging Green Button Go to advance their portfolio. But what does lab automation entail? Historically, when we think about lab automation, the first thing that may come to mind is the large robotic arm, the conveyor belt. But automation is more than just the physical. Automation also encompasses the logical aspects. So this is the workflow, the sample types, the business rules. It's the way scientists will actually interact with the automation. In addition to that, and maybe the most important aspect of lab automation is the data. And the data aspect that ultimately is why we're doing the science. And so what does this actually look like in the laboratory? And some of you have seen this workflow before, or maybe you actually do this workflow on a daily basis. But if we started where we had a scientist that actually interacted with some sort of sample storage to grab dry compounds, they would then take those compounds over to a balance where they'd weigh out a certain amount of dry material into a barcoded vial where they'd capture the weight and the barcoded vial ID. They'd walk those vials over where they would add or pipette a solvent to solubilize those to a desired concentration, 10 millimolar DMSO stock solution, and then they take those compounds or samples over to a work cell where that work cell is going to transfer those samples from vials into plate format, add some reagents, and now we have a compound-ready plate that is walked over to, say, a liquid handler, where it's met with a plate of cells that have been previously cultured on another system or in another lab. The liquid handler doses the compounds onto the cells, reagents are added, there's some incubation, and finally, that assay plate is taken over to a analysis station where some sort of endpoint analysis is done, whether that's a multimode reader, high content imaging, flow cytometer, and out of that comes the experimental data. But in addition to this workflow that encompasses the logical, the physical, and the data aspects, there's also the need for human interactions where we have consumables and reagents and buffer blends that need to be at each step in this process. And out of each step, there's the generation of waste materials that also has to be collected and discarded. And when you look at this, you quickly realize that there needs to be a way to manage the laboratory, or better yet, orchestrate the laboratory. And it's not just about the incoming sample data or the experimental results. Lab automation is also about the capture, contextualization, and the harmonization of all of the data that is generated along the way. And it's the metadata, the humidity and the temperature in the laboratory, the lot of tips that were being used, or the type of seal that was used to seal a plate. All of that information is extremely important in being able to provide AI, machine learning, retro-experimental analysis tools. And so what is lab orchestration? Our definition is it is the management of workflows by harmonizing processes, people, data, business rules, and materials to achieve desired results. And as we know, the scientific process is not a linear one. It's a cyclical one that's based upon the learnings and the optimization to enable better predictability in the future. And so in 2022, Biosero launched Green Button Go Orchestrator to the market for the first time. Our Green Button Go Orchestrator was a software suite that was comprised of 8 applications shown here, involved in the planning, scheduling, execution, and optimization of those end-to-end workflows within the laboratory. The architecture that many of you have seen before is one where it's a modular and scalable architecture where you're able to mix and match the different applications. And at the heart of the orchestrated solution is this application called data services. It is the connectivity hub, both with the internal and external world. And what we found over the last 3 years, as we've implemented over 100 orchestrated solutions across a variety of different laboratories, we've found that there's really 3 common lab orchestration blueprints in any combination of those. And what it entails is, in each one of these, you have digital connectivity with a differing mode of physical connectivity. And so we've enabled islands of automation that are digitally connected, but their scientists are moving materials from one island to another. We've enabled islands of automation digitally connected that a mobile robot is the mode of connection of transporting materials from one work cell or instrument to another. And finally, we've enabled a track-based connectivity model where we're using some sort of physical tethering system to connect those different work cells along with that digital connectivity. And over this time, we've seen that the 8 applications that are part of our Orchestrator software suite, in most cases, many of those applications were actually leveraged as part of that solution. And so in 2025, we launched 2 core products of Biosero, our Green Button Go Scheduler flagship product, as well as a simplified Green Button Go Orchestrator, which encompasses many of those applications that we've seen to be successful in the implementations. And so our new Green Button Go Scheduler has been upgraded with the API connectivity through Data Services Lite. So as I showed the original architecture, Data Services is that connectivity hub, both to the internal and external world. Now Data Services Lite comes as a part of our Green Button Go Scheduler product. And with this, you still have the traditional drag and drop workflow configuration. You have the native programming engine. You also have access to hundreds of device drivers, and you have that real-time monitoring and troubleshooting as a part of the software. But now you have, from the beginning, connectivity into the data services application, which allows for consistent API connectivity and a seamless transition into the evolution of the Orchestrator product. And our Green Button Go Orchestrator has now been launched as a software suite, but it's bundled for success. And what it entails is many of those applications that have been so successful in delivering the orchestrated lab. So for those users who may start with a single work cell, Green Button Go Scheduler on it, they now have the ability to seamlessly move into the completely orchestrated lab, where you now have access to our workflow, our Green Button Go Workflow Designer, our Green Button Go Workflow Conductor, our data services, as well as Green Button Go Transportation Manager, being able to connect with multiple instances of Green Button Go Scheduler. And for those laboratories that also want to connect standalone instruments or digitize manual tasks, you now have the ability to add on those additional applications of our Green Button Go Instrument Agent or Green Button Go Lab Experience. And so what Biosero is enabling is a modular approach to more automation, more connectivity, and more flexibility. And when you look at this system on the screen, historically, this would have been one monolithic system that was only as good as its weakest link. If an instrument went down, potentially it took the whole system offline. Today, Biosero is enabling this through being able to have independent, autonomous work cells that are running under their own instance of Green Button Go Scheduler, all of which are incorporated under the Green Button Go Orchestrator product, which is allowing labs to build up their automation footprint over time as their budget or portfolio enables them to do so. It also allows for the ability to ensure stability or development time, where you can simply take one of the Green Button Go Scheduler work cells offline for PMs, for development, while the rest of the system continues to stay in motion. And so we are approaching this with a modular approach, with now a focus on control at your convenience. And with that, we launched 2 additional products. We have our new Biosero Portal and our Green Button Go GoCare subscription. The Biosero Portal is a new tool, marketplace. You can think of it much like an online app store. And what this is allowing is control at your fingertips, where you now have the ability to manage 24/7 your Green Button Go licenses, your user management, your driver marketplace, so you can go in and access all the drivers that are available and have the ability to exchange those. We also are now offering our training, on-demand training tutorials, as well as you have the ability to access our support channel. And this not only incorporates what's in your lab or campus, but this allows those users who are multi-site to manage those different licenses anywhere across the world. In addition to that, we are launching our Green Button Go GoCare subscription offering, which is really about enhancing the usability and maximizing the return on investment. As a part of this, you have the ability to view and manage your licenses, user subscriptions in a 24/7 manner. You have access to virtual troubleshooting. You have access to on-demand driver exchanges. So as an instrument changes, you can go in and pull that down. You have the ability to tap into that on-demand training at any time of the day, wherever you're located. And you also have real-time access to software upgrades, patches, and driver updates whenever you're ready for that. And in conclusion, Biosero is supporting scientists with a strong foundation. We're developing a platform to unify the physical, the logical, and the digital elements that go into making the orchestrated lab. We're offering flexible tools that support you where you're at today and give you that real-time access to control your environment when you need it. We continue to deliver device connectivity through our Green Button Go scheduler and tapping into the driver library of hundreds of devices, and we're enabling digital connectivity through Green Button Go Orchestrator and our informatics adapters, which allow you now to seamlessly connect into the data tools and the software systems that you're using in your laboratory. And to bring this, what this enables is it enables the ability for scientists to unlock the potential within their laboratory. And so what you're seeing here is all of these elements coming together to enable an AI-driven design, make, test, analyze, discovery platform, which is built by Biosero and powered by Green Button Go software. In this, we've integrated over 20 different vendors from both hardware and software, hundreds of devices across 20 different platforms, all to orchestrate that laboratory and enable end-to-end workflows that are going to provide answers to some of the most difficult questions faced today. And with that, I want to thank you again for joining us, and I want to also thank our 4 speakers today that are going to tell you a little more about bringing their scientific vision to life in partnership with Biosero across a variety of different market segments and areas of scientific application. Thank you very much.
Hello. So I'm Susie Cox, and I work at Corteva Agriscience. The story I'm telling you about today is a little bit about my former team. Currently, I lead our compound repository and screening team at Corteva, which is taking care of the management of all of our active ingredients in addition to the bioefficacy collection data. Previously, we were working on automating formulation development for agrochemicals, which is a highly challenging task. And I'm hoping to provide you kind of some insight in there and the importance of our collaboration with Biosero to make that possible. So with that, Corteva does have a rich history. So we are kind of the spinoff of Dow and DuPont. So we have 140 years plus of background. We are a global company, and we need to help farmers across the entire world. And so when we look at farming in the US versus China versus Brazil, it all looks very different. We need to be able to have a product that can be in a drone, a product that can be in a backpack, a product that can be hand cast, or maybe that more traditional farming that's in the Midwest of being in a large spray tank. And so there's a lot we really need to think about in that space. And we do have 140 R&D facilities worldwide. And there should be a map of the world behind that, but it is missing in action. So that, just to kind of get across to that. In terms of how we approach formulation development, one of our core values is sustainability. And so one way we can do that is through our active ingredients. And so this is an example of Rinskor, which won the Green Chemistry Award. You can see, just visually speaking, how much less material it's taking to treat the same amount of crops. And so within that space, the untreated rice is that kind of, you're seeing the fuzziness, that's the weeds in there, versus the treated rice to your left. And so this is just one handle that we can have on that. The other way we can look at this is through formulation and packaging innovations, and this is very much an eye chart. I won't go into this, but we've really had a huge focus in this space. So let's talk about automation. Automation and agrochemical, what is our motivation for automating product development? What's our foundational choices that it takes to do that? Specifically, our collaboration with Biosero was to do characterization work. And then finally, the case study with some data and some numbers as to kind of what does that look like from a scientific perspective. So from a formulations standpoint, the importance of formulating your active ingredient is shown very well in this video. And so that clear droplet's just water, and you can see it bouncing off the leaf, versus our formulated product is gonna really stick to the leaf and spread out. So you can get higher efficacy if you're getting it to its active site. So for example, foliar uptake would be the case that we would want. So this is our Inatrek IQ4 technology. And to be able to do that, to make your formulation do that, you need to think about a lot of things. And so we always start with our grower in mind. Each one of these circles in this cycle is equally important. So it's certainly a balance of our application, our stability, our efficacy, our cost, working with our supply chain partners, and certainly making sure it's safe for those who are using the land, right? Within a formulation, there's a number of different inerts. And so I'm just putting this here to kind of, for you to appreciate the number of variables that a formulation scientist needs to be able to tune to get that product to do what we need it to do in the spray tank, in the silo, in the drone, in the backpack, right? So there's a number of different components that allow us to get into that space. So when we took time to say, what exactly are we automating? Because there were so many choices, we took a step back and said, let's look at our heritage data. What are we testing? So historically, we always found there were these top 6 tests, with emulsion stability being the highest one, and I'll give you an example of what that looks like. So we said, okay, we need to focus our characterization on these tests. Now we have different tests for different formulation types. There are a lot of formulation types out there, so I'm not going to go into that, but this shows you the different ones that we sell. So we picked out which is our top sellers. So our top 3 are water dispersible granules, which is a solid product, suspension concentrates, which is a heterogeneous product, a lot like paint. So you have a crystal supported in an aqueous medium. And then an emulsifiable concentrate, which is going to be your active dissolved in some sort of sustainable solvent that then emulsifies when it hits water. So that's where we knew how to focus, is really just taking a step back and looking at the data. When we talk about these tests, these are not automation friendly. That is the reason for this slide, is to show that to you. So emulsion stability is done in a 100 ml conical tube. And one example is when it's at room temperature versus 5 centigrade. So at 5 centigrade, this is failing. And it's very important for us to understand that, because this formulation could not go to Canada. Right? But maybe it could go to Brazil. So these are some things that we really need to understand across the board. A formulation scientist will take a look at this, and it's not a red formulation, that's the redness of the tube, but it's completely a broken formulation. A formulation would say, oh, there's creaming, I need to adjust this thing. Oh, there's oil, I need to do this. And so these failure modes are important for us to be able to both capture and then share. The other things we do are persistent foaming. You don't want any foam in a recirculating tank, because the foam would come out the top. This is a 250 ml glass conical tube, or graduated cylinder. We do a lot of rheology, a lot of particle size. We need to make sure we're not going to clog spray nozzles, because that can be highly expensive for the farmer to clean out. And then of course we do this wet sieve test, which is both showing us particle size and agglomeration. Once again, please take a moment and think the challenges of automating the things on here. So we really had to take a step back and say, okay, these are the most important. How are we going to do that? Then we need to think about our formulations. What's the same? What's different? What's most important? And so this is an image of each of the 3 top ones I told you about. They all have solid components. They all have liquid components. They all need to be diluted into different water types. How are we going to dilute that? How are we going to transfer a solid versus a liquid versus something viscous, right? So these are the core foundational things that we had to think about when we were creating a system that could characterize on such a complex plane. In terms of vial type, and this is just as a hill I will die on, is the most standardized vial and plate you can get into is so important. And so we needed to think about compatibility with plastic versus glass. We needed to think about the size. Maybe our solids need to have a larger vial just because of bulk densities, things about viscosities and the like. So we took a lot of time thinking about that. And when we had collected all this information, we started talking to Biosero about what could we build together that would be able to do this, because we recognized that no piece of equipment existed that could do those tests. So that is something we needed to automate, but we could keep certain things standardized. So this brings me to our FORCE team. So the Formulation Optimization Research Capability Enablement. You might see the heavy Star Wars theme. So we found it very big deal that we completed FAT on May the 4th. We have never forgotten that. And in terms of the different features that are on there, we do have quite a bit of imaging work that we've had to do in terms of to allow us to look at that solubility screening, dilution screening, and the like. We also have assay development work on there. So we do have an HPLC. We do some understanding of water characterization. Is there crystallization? How are we doing particle size and the like? So unfortunately, I can't go into the details, but what I can tell you is there are 2 different robotic arms on here. So we have a 6-axis Stably arm, and then we have the PF4000 arm, or 400 arm. And this is allowing us to be able to run all these tests asynchronously so that we don't have to wait for one thing to complete before the next one continues on. And so I can't show you the inside of our lab, so I went to ChatGPT to help me explain what we are doing in our lab. And so we have broken this down into sample preparation, so we have 1 piece of equipment for that. I'm not talking about that today. We have our automated sample characterization system, so everything from automated sample prep gets moved over to our automated sample characterization. And then we have our automated rheology platform. And so just kind of in terms of looking at that kind of that Biosero system and where it fits, right? So really talking again about the importance of having that scheduling software to allow us to run all these different tests at once. The other thing I want to touch on is this has allowed us to really standardize how we operate. Scientists are wonderful. They also like to be unique, which can make it very challenging when you're trying to do some sort of data projects or machine learning and optimization through that. And so this is an opportunity for us to have standardization across the board for all of our labs. And so let's talk about the data and the science and an example of how we're doing this. So once again, we're going back to that emulsifiable concentrate. And so when we dilute an emulsifiable concentrate, I mentioned it does emulsify. This bloom takes a lot of effort in the balance of those components. It also needs to do this in multiple different water types and then stay stable for up to 24 hours or more. Traditionally, our scientists would think through this 3-dimensionally by utilizing a ternary plot. Because you only have so much time in a day, they might have to scatter the points on this plot, right? And try to get the largest design space. And so within that, they then find what's good and what's bad. And so then they then refocus more on the good, remake a new triangle, and continue. What this leaves out is it leaves out the nuances of learning from what's bad. If you think about how you record an experiment, how much time do you spend recording the bad data, right? It is all good data. And so what automation allowed us to do is we turned to spectroscopy, and we had to train it to say what's good and what's bad spectroscopically. And so we spent some time in that space, but we were able to fill out that full triangle. And one thing I want to point out is there's actually a better good. The better good is when you have more green, because that means it's less likely to break with slight changes in your formulation. So we learned that. we also learned what to avoid in the future. And so that way, our other scientists wouldn't necessarily go back to that first, and they could try better things moving forward. We have this in sort of a stepwise approach. So we have a fast screen, and then we have a more focused screen. And so I always say we are creating that fuzzy image and then bringing it into focus. And so the trade-offs, right, are productivity versus resolution. So our traditional technique to run 96 samples, so we went 96 well plate, takes about 2 weeks, and it's gonna generate over 10 liters of hazardous waste. Using this initial very quick screen, we're able to do this in 30 minutes and generate very small amounts of hazardous waste. And then we can take the best of those and put them through an intermediate screen before taking them to our traditional technique. Once again, this does go back to that piece with our team that we are very much focused on the data management. So that's another thing that Biosero has allowed us to do, is by having a standardized way of operating, we can have a standardized way of managing our data. So we have a centralized database for all of the work that's done, so you can track from the beginning to the end and beyond. This is then used to have standardized data interpretation to allow our scientists to kind of start really understanding the plots across the teams. And then finally fed into a machine learning model where we can create roadmaps for future formulation development. And so that's been a really exciting outcome of this project, is just learning from the past in a really big way. So how we do it, I am representing a much larger team. So we have a very incredible team that's not just mine, but all the teams we collaborate with. And so together we are putting this all together. We need those data scientists, we need those engineers, we need our biologists and the like to be able to do this. We also find the importance of having fun. So I will share this very briefly. This was taken at the end of our FAT. And it does have a purpose. I'll tell you here in a second. So this is just a reminder that these arms work asynchronously, right? And so they can move on their own time. But it's really important for us to kind of keep it light and keep it bright, because automation is challenging, right? And so that's been a big piece of this, is staying open to new ways of working in everything we do. So with that, I'd just like to say thank you, and a big thank you to our Corteva partners in the room, because they've been involved in this as well, and they've done a ton of work. So thank you very much.
Okay. How's it sound? Good? Okay. Hi, everybody. My name is Bill with Merck. Today I'm going to be talking about automating traditional pharmacokinetic assays in incubator lab. Incubator lab, it's not a lab that's set to 37C with 5% CO2. It is incubator in the technological sense. It's a place to bring in and test new technology before we ship it out. That was a joke. Oh, too far away. Okay. Oh, wrong way. All right, so just a little bit about myself. I have been at Merck for 2 years. I started out as a vendor. So in about 2012, we got into lab automation, where we would actually OEM Thermo Fisher Momentum and Green Button Go through molecular devices. Then I moved to Beckman Coulter, where I did some solutioning for automation with the SAMI platform. So I've been in automation for a pretty long time. At Merck, I started out as an associate principal scientist in robotics, and I've just recently moved to IT, which we call the dark side. I'm not going to go through my interests, but I do have a shameless plug. If you are in the Philadelphia area, I own a small bar about 45 minutes outside, and you guys should stop by. It's a good bar. All right, so what's going on at Merck as far as their investment strategy? So this presentation is pretty high level, and I'm going to go pretty fast, but I'll be available for questions afterwards. So at Merck right now, just in the landscape in general, technology is advancing incredibly fast. It always has, but it seems like it's faster now. I mean, if you look, every news article that comes out talks about AI this, machine learning that. And just recently, the current administration launched a Stargate, which unfortunately isn't the Stargate I thought it would be. It's a data center. So that technology is advancing very quickly, and our industry is historically slow at adopting technology. But Merck has recognized that the future of science is going to be more tightly coupled with technology, from your plate washers to your software to your Excel macro that generates your heat map, right? So to that extent, they've started a program called LROP. LROP stands for Long-Term Research Operating Plan, and what that does is it provides funding sources for people to do cool projects. And these projects have very specific purposes. They're to support our ever-growing pipeline. The pipeline is growing in size and in complexity as we bring in lots of new modalities and things like that. So this funding program is designed to help you support that pipeline, not today, not tomorrow, but it has a 10-year look, right? So we're really looking at modern architecture and things that are going to be around for a long period of time. So when we went to the LROP program to get our funding, we went with the goal, the goal, which was to decrease the cycle time for drug discovery. So that includes lead, ID, optimization, and candidate selection. That really is going from target identification to preclinical candidate. That's where we sit in the research operating plan. The how of we were going to do that is we're going to modernize our PK and our toxicity assays. And those were historically more manual assays. There were some automated Hamiltons, but that was kind of an aging architecture. So modernization comes in 3 flavors with this project. It comes in lab automation, which is why a lot of us are here, data automation or digital automation, and new capability. So we kind of hit all 3 of those with this incubator lab. So the where is the incubator lab. So this lab is supposed to be a space that helps de-risk automation for other departments. So we have all kinds of cool new technology in there. And we come in, we'll POC assays. So a lot of our goals are proof of concepts for automating an assay. And then we will take the budget that it took to build them, the preliminary POC data, and we can deploy those to other parts of our company. Essentially, what we're trying to prevent is that, 2,000-pound paperweight that you sometimes see. So just a little bit about the incubator lab. So the incubator lab consists of 3 large Green Button Go powered platforms. We have a sample plating and prep platform, which allows us to do both acoustic and tip-based assay-ready plate creation. That platform feeds our general automation platform, which is a large, broad endpoint platform. So it's designed to be able to handle a lot of different endpoints. Currently, it can do high content imaging, plate reading, and which I'll talk about a little bit more, the echo mass spec. We also have what we call the vending machine ELISA platform. It is a small throughput MSD SpectraMax platform that customers, or I call them customers, or partners can come up and do design of experiment on. We have an integrated Hamilton for some cell line maintenance work. That's for our toxicity models. And we've brought in some new technology, which I'll hit on a little bit later with the NovoSeq X Plus and again, the 7600. So some of the key features of the lab is large, right? It's a double lab space. It's got 8-foot tall ceilings, which is important because all of us that have done lab automation knows we often come into that 18 inches from the sprinkler head. So the sprinkler head is high enough that the robot arm doesn't touch it. We also have future-proofed the lab with the utility troughs above. And I'm just going through this so you can kind of get an idea how to future-proof your own labs. We have 50% extra power, configurable gases, vents that you can, if you have potent compounds, you can connect anything to your vent to get it out, and tons of data support. The casework in this lab, which is the fancy word for the benches and the robots, are all on wheels, so we can move them around and reconfigure the lab when we bring in new capabilities. One of the things that surprised me the most was the benefit we got from that glass lab connected to the floor, right? It has open touchdown space in it, and it's connected to the lab. You can see in and watch the instruments, and it really has reduced the barrier of entry for our scientific partners to come in and POC a lot of those new assays in this lab. They sit there, they can do their work, watch the robots run when the grid lights are green, and they run in when the lights turn red. So what happened in 2024? So we were pretty successful. We were able to hit about 90% of the milestones we set out to the business as far as proof of concepting assays. We got to the point where, we started moving towards deployment. So again, the idea of the incubator lab is to take pieces of technology and deploy them out. So we're in the process right now of deploying 8 more systems. I think 6 of them are Green Button Go platforms that are going to go in West Point, San Francisco, and Boston. And I think one of the most important things that we got out of this experiment was program support for peptide stability. So, the lab was turned over to us in January, and with the help of Biosero, we were able to have the entire lab built up and running by February. March, we were science ready, and we were doing program support for peptide stability by the summer, to the extent that the chemists now have doubled their orders for the peptide stability assay. So pretty successful. I'm not going to go into too much detail with all different POCs. If you want to ask me later, you can. But some of the cool capabilities we have are the next-gen sequencing transcriptomics. So that's really going to speed up our turnaround on the toxicity assays. And again, the Echo 7600 Plus. Digital automation, real quick, I'll talk about. We spun up an S3 bucket to capture AWS, so Amazon Web Service S3 bucket, to capture all of our process data. So all of the liquid handling files, dispenser files, all those things get put in a central repository so that we can analyze them and parse them at a later date. We also started to spin up an EC2 instance to host some of our parsers that I'll talk about more. And that really kind of I'm going to piece this all together about how Green Button Go helps us make error-proof runs in that lab. And if anyone's a mass spec person here, we're using something called Onero to send our data out and do a lot of the analysis. And that's taken us from about 10,000 clicks down to a couple hundred for analysis. So we're really working towards the ability to do review by exception only for stability data. All right, so why Biosero for this lab? Quickly, I just kind of want to talk about the team. I think Susie mentioned the importance of a good team, and I think the team is a function of the success. We have 4 dedicated automation engineers, 3 dedicated data scientists, and 1 dedicated IT guru who is always busy. On top of that, we have about 7 to 8 really good scientific partners. So we're a DIY kind of automation engineering shop. And in the spirit of the incubator lab, it had to be as flexible as possible because we don't know what we're going to need to do in 10 years to support the various pipeline. 5 minutes, okay. I'm going to go fast. So we went with Green Button Go for several reasons. As a company, Green Button Go Biosero is software first, and they're truly our vendor agnostic. As a software, you have the ability to control and modify anything. So it really gives us the power and the flexibility to write the programs we need. I like the DLL architecture. I can get into that later offline. And then I'm going to talk a little bit more about the customizable UI and the open C# scripting interface. So real quick, one of my goals is to always democratize automation so that anybody can run it. And with Green Button Go, I can do that with the UI interface and the scripting behind the scene. So what we did here is we built wizards for every application. So when a scientist walks up, they just go through step by step on the UI to load the system and set the parameters. We parameterize most of the runs so that if you need to do things like modify, add cofactor, add a wash, skip something, change incubation times, any of those DOE type of parameters, the analyst can use those by typing things in here or selecting checkboxes. And what that does is it kind of removes them and it abstracts them from the method, and it makes it just a lot easier for them to run. And I'm going to tell you that with the UI and the scripting interface behind, we've had scientists that had 0 automation experience running peptide stability within 2 weeks. And after we modified some of the code to error-proof it, they're running error-free for probably since, I would say, the summer. So it's been pretty successful. Behind the scenes with the scripting interface, what we have is the AWS-hosted parser will take the LN master instruction file, so it comes from our lab notebook, generates all of the instrument instruction files. So for this particular case, it's the Dragonfly file, Hamilton, and all the sequence file for the mass spec. And it automatically puts those in a central repository and points the program at the correct file. Those were our 2 biggest errors. They would load the storage wrong, and they would pick the wrong files. by scripting this out and giving them the UI, we took all of that out of the equation. So now they just type the notebook in and everything is automatic. The other thing we can do is with the scripting, we do some infographic loading. So we generate some simple HTML to kind of show the users where to load the matrices and things like that. And again, what we're doing in the term, it's actually a Japanese term, I think, pokeyoke. We're trying to just take all of the error out of the process. And we're doing that with Green Button Go with the UI and the scripting interface. Just talked a little bit about the team, but I also want to talk about the importance of the scientific partners. So anyone in here that's a scientist that's don't really consider yourself an automation engineer, but you're on the periphery, you guys are the ones that are driving the success. So our scientific partners from the director levels that are pushing adoption to the analyst levels that are coming to me and saying like, I don't like this part about it, can you change it? Keep doing that, keep driving that improvement, and that really will lead to success. And then finally, I have to thank the vendors because I think Susie was thanking, Biosero. The vendor relationship and vendor management is so important, and we wouldn't be where we were if we didn't have solid vendor relationships. How'd I do on time? Good? All right, thank you.
Okay. Hello. Thank you very much. My name is Ichiji Namatame. I'm in charge of automation development and management. Today, I will be talking about automation for iPS cell drug discovery at Astellas Pharma, which is aimed at the global expansion of orchestration involving humans, robots, and AI. This slide shows today's agenda. I will talk about 3 topics. First, the background of our automation system installation. Next, humans, robots, and AI working together to solve issues and achieve global expansion. And finally, 2 blueprints for the global expansion of orchestration. This slide shows Astellas Modality Research Centers. The term modality refers to the methods of drug discovery technology. The Discovery Intelligence Center, located in Tsukuba, Japan, where I work, focuses on small molecules and antibodies. Astellas is also globally involved in various modality drug discovery efforts, including gene editing at Universal Cells in Seattle, gene therapy at Astellas Gene Therapies in South San Francisco, next-generation cancer cell therapy at Zyphos, and stem cell and differentiated cell production technologies at ARM in Boston. This slide shows the applications and challenges of using iPS cells in drug discovery. One key application is their role in cell therapy. Our particular focus, however, is on iPS cells as in vitro human disease models, highlighting their potential to improve clinical predictability in drug efficacy and toxicity evaluations. We expect that this approach will streamline the assessment process from small animals to humans and thereby accelerate drug development. iPS cells are also important for target discovery and in understanding disease mechanisms. However, they come with a number of challenges. These include low reproducibility due to individual and experimental variation, the need for extensive maintenance in long-term culture, such as daily media changes and continuous morphological observation, difficulties in transferring skilled experimental techniques, and challenges in quantifying 3-dimensional cellular changes during multi-sample screening. In other words, the issues related to the use of iPS cells in drug discovery include the need for advanced skills, time, patience, and relentless effort. The solution involves the use of automation and AI technologies. Specifically, we have installed Mahoro, a robot for cell preparation, and the screening station for screening and assays. Astellas strategies to address these challenges is called Mahoraba. Mahoraba is our unique platform which consists of two island automations, namely the Labodroid Mahoro, which is responsible for preparing iPS cells, and the screening station, which handles screening and assays. This strategy fosters collaboration among researchers, robots, and AI. All related papers have been published and you can access them using the QR code provided. The second paper regarding the screening station was published last November and is featured in this SLAS 2025 poster 1263D. So please take a look. Our company website shows a drug discovery platform integrating humans, AI, and robots. You can also access these using the QR code provided. Oh, sorry. Sorry. That's me at right. This slide shows a movie about Mahoro or its layout. Mahoro can automatically execute IPS media changes and passages and thereby ensure a stable supply of many types of high-quality cells. As a result, cell preparation that requires advanced skills has been automated by Mahoro, reducing the time, patience, and effort needed. Under collaboration between Astellas and the Center for iPS Cell Research and Application, CYRA, at Kyoto University, we transferred the skilled manual protocol from CYRA to Mahoro at Astellas Tsukuba using skeletal muscle iPS cells derived from patients with FSHD. We automated the differentiation process and immunostaining, resulting in success in the initial tests. This initiative serves as a successful example of human-robot collaboration in the automation protocol transfer of cell preparations between distant institutions in Japan. This slide shows the layout of screening station. The screening station is an integrated system consists of 18 types of equipment, including a Novocyte flow cytometer, CQ1 image cytometer, Scale 48W automated CO2 incubator, 405LS washer, and Biomech i7 liquid handler. The screening station was integrated by Roche Life Science, a vendor in Japan, and all scheduling was executed using Green Button Go in Biosero. The result is that the screening station has automated screening and assays that require advanced skills, reducing the time, patience, and effort needed. The screening station has 3 basic workflows as shown in the movies here. Media change, live cell time-lapse imaging, and immunostaining, and assays. Several robots transfer cell plates and labware, allowing each workflow to be executed. The movie of left side is media change of cell plate using Biomech i7. The movie of right side is live cell time-lapse imaging, is using CQ1 and immunostaining assay using 405LS. Okay. Similar to the example of Mahoro, or the collaborative research between Astellas and CYRA at Kyoto University also includes integration of the same devices into the Tsukuba screening station, including the Biomech washer 405LS and CQ1. Semi-automated media changes, time-lapse imaging, and immunostaining were performed with manual labware transport in CYRA. We transferred the program from CYRA to the screening station in Tsukuba, enabling labware transport to be executed fully automatically, and this also succeeded in the initial test using FSHD iPS cells. This is a successful example of the transfer of an assay automation program from semi-automatic to fully automatic through human-robot collaboration within Japan. This slide shows the time-lapse analysis of cell density and differentiation using automated imaging and deep learning image analysis techniques. We have utilized AI to check cell quality and quantity. Cell plates are transported from the CO2 incubator to the imager CQ1, where data is automatically transferred to the server and analyzed by AI. We used deep learning techniques from Yokogawa Electric to create training data based on the skilled experience of advanced researchers in analyzing the cell density and differentiation of FSHD iPS cells. This is a successful example of live cell time-lapse imaging and AI analysis through human-robot AI collaboration. Mahoraba can perform protocol creation, assays, imaging, and analysis by remotely accessing the laboratory from a distant location after the simple setting up of reagents and labware. We can start experiments and make troubleshooting decisions from a cell phone or laptop under Astellas network security. The ability to operate the system remotely has enabled the further global expansion of Mahoraba. As mentioned, we have successfully expanded Mahoraba within Japan and implemented a similar system at Syra outside Astellas, achieving program transfer. Additionally, by utilizing internet-based social media to share protocols and know-how, the community for screening station number 1 has grown to about 150 members with more than 10 heavy users. Since our needs extended beyond the capacity of screening station number 1, we made the decision to install screening station number 2 as an advanced model in Japan, and it is now under construction. Furthermore, we installed screening station number 3, a copy of screening station number 1 at South San Francisco last year, allowing for program transfer so that the same experiment can be conducted not only in Japan, but also in the United States. In other words, we have executed the global expansion of Mahoraba. This slide shows one of the blueprints. Our proposed orchestration of humans, robots, and AI involves building an automatic feedback system that connects AI data to robot operations based on human ideas. For example, the calculated values from AI analysis are fed back to the liquid handler and the robot movements by the GBG orchestrator, and so on. This slide shows another blueprint. It aims to accelerate drug discovery through the global sharing of data and digital workflows centered in Tsukuba, Japan, as well as through internal expansion in the US and the external expansion of orchestration involving humans, robots, and AI. In conclusion, today I have introduced to you some of the challenges we have faced and how we are overcoming them. These achievements have been made possible through the cooperation of many vendors, including Biosero and Roche Life Science, as well as a member of Kyoto University Syra and Astellas, and stakeholder from our group companies, Astellas Gene Therapies and Zyphos. I sincerely appreciate all the support we have received from everyone. Thank you all. ありがとうございました。
Uh-oh, clearing the room out. All right, my name is Julia. I am the supervisor of the Neuroimmunology Laboratory at Mayo Clinic in Rochester, Minnesota. I'm here today to talk about our experience automating laboratory medicine. So if you're anything like me, before I started working in the neuroimmunology lab, I had no idea what that meant. So we are a 24/7 laboratory that aids in the diagnosis of autoimmune diseases, specifically those that are affecting the brain, nerves, or muscles. So some of the more common diseases you might have heard of are epilepsy, dementia, encephalopathy, and even type 1 diabetes falls into that world of testing. And oftentimes we will end up diagnosing unsuspected cancers, some of the more common ones being like small cell lung cancer or even testicular cancer. In order to diagnose these tests for our patients, we end up using multiple different testing platforms. So we use immunoprecipitation, immunofluorescence assays, Western blots, ELISAs, chemiluminescent assays, flow cytometry assays. RT-Quick is our newest testing platform in the lab, and then we also have some live cell assays that we use. I would say about 98% of the testing that we do in our laboratory is lab developed test, meaning that people at Mayo Clinic actually developed our assays, so we can't just buy a kit and purchase our QC or even get proficiency testing samples from CAP like a lot of other labs. So we have to do a lot of that all on our own, and we create our own SOPs. So automating stuff for us was rather complicated. So as we were trying to decide how we were going to keep up with the growth, we were sitting here in 2019 when we started, looking at our year-over-year growth rates. I have a little chart over there for you to take a peek at if you'd like, but when we were taking a look at this, we had a 13% year-over-year growth rate and double-digit year-over-year growth rates up until 2020, where we dropped down to about 7 or 8% growth. And then last year, we finished with an 18% year-over-year growth rate. So our testing needs were growing faster than we could possibly expand because we don't have enough space in our laboratory. And I don't know how many of you have had to try to hire individuals recently, but finding qualified staff members has been rather difficult. And then in the world of science, our testing is relatively new, so the discovery pipeline is rather robust. We currently have 22 tests in our development queue, and I think if the laboratory clinical staff could take on more tests, they'd have those all live now. So as I said, we started this journey in 2019. We partnered with Hamilton and then another well-recognized vendor at the beginning who does track systems that we decided not to go with due to budgetary reasons later on. But as you guys are all aware, in 2020, COVID happened. So during that time, we had to shut down our projects so that Mayo Clinic could focus more on doing that COVID testing for patients around the world. And during that time, we actually got to work with the FDA, and our laboratory did the neutralizing convalescent plasma assay to prove that getting that convalescent plasma would help treat COVID-19. So that was pretty exciting. But to the end of 2020, when our project got resumed, and we realized that there was a lot of staff turnover on all sides. So from the Mayo Clinic side, when this project started, I was in the assistant supervisor position, but the assistant supervisor, supervisor, operations manager, operations administrator, associate administrator, and the department chair had all changed over due to promotions in other places. Thankfully, I went on to the supervisor position, so I was very familiar with the role, but Hamilton also had some staff turnover. So their pre-sales individual and the programmer who had, between the two of them, figured out how we were going to move forward, both left the company. And basically, we had to start over with a new staff on that front and then come up with a new plan. So there were some other struggles, like no matter what happened and how we tried to fit our automation line into our laboratory space, there was a pillar, a big giant cement pillar that no matter how many times we asked, Mayo Clinic said we could not take down since we're on the 7th floor of a building. And then also trying to raise the roof, which they did let us do that for our Verso into the laboratory, which is an automated storage space. But Q then, like I said, the Hamilton individuals left, but the remaining members at Hamilton brought us to Biosero. So the two companies partnered together to help us reimagine our automation line. and helped us create what we're working with right now. And current state, we are in the process of proving the science behind all of our assays, making sure that everything is up to the standards that Mayo Clinic has. And we are getting ready to do full path verification for our automation line. So this really was a collaborative effort. Many stakeholders from the Mayo Clinic, including all of our internal lab staff, so specialists, management, we have a project management team, and then we also created our own internal small application for our laboratory. It's called NERDS. We're pretty proud of the name, but it's the Neuroimmunology Electronic Records and Document System. So that allows us to create API interfaces between Biosero and our LIS soft. And then Hamilton played a huge role. They are providing all of the equipment on each of our work cells. So each one of those work cells there, it consists of a Hamilton star and then an integration cart that's filled with third-party components so that we can do our testing. So when the full line is in there, we'll end up with 5 ELISA systems, 4 flow cytometry systems, an IPA system, a sample processing system, and then 1 automated storage system in the Verso. So then Biosero, they're of course part of this project, that's why I'm here today. But Green Button Go, their application, is powering each one of the individual work cells. And like I said, that work cell is the Hamilton star and the integrated third-party components. And then the orchestrator software is coordinating all of those systems together so that they work as a cohesive unit. And that's what really is bringing everything together. And then we do have a track system. It's actually a Rochester company, Alliant Technologies Group. We call them ATG. And they're doing the conveyor, and there's little elevators everywhere. So I just wanted to give you a quick look inside each of these integration carts. They are set up differently, like I mentioned. So for the ELISA system, any samples that come into our lab and go onto the automation line, they start and finish on there. So it's kind of hard to see. It looked a little bit bigger on my computer there, but on both systems, you'll note that there is a sealer and a peeler. So if you have any incubations, we seal those up so that there's no patient carryover or contamination and no evaporation during the testing. Each of those systems actually has an incubator shaker system. There's plate washers on both systems. The ELISA system has a plate reader, so we get that data. And then each system has a capper decapper that not only can cap and decap samples, but it also takes an image of the samples as they come in and make sure that the appropriate samples are on each system before we start testing. So what are we hoping to accomplish? We are hoping to see some improved quality. As those of you who work anywhere know, anytime you have a human doing a job, there are always going to be things that happen, just that human error rate. So we are automating a lot of that upfront processing to get our samples into a 96-well format using the help of all this equipment here. We're hoping to reduce our turnaround time. So each sample, when it comes into our laboratory, is in a 5 ml tube. And with the help of Hamilton and Biosero, we are now taking that 5 ml tube and we're going to make 1 ml aliquots for each sample. Those samples are matrix tubes. And so we're hoping that we can test on multiple platforms at the same time, making it quite a bit faster for our patients to get results. Reduction in FTE through avoidance. That's always kind of a touchy subject. The reason I say through avoidance is because we're not firing anybody in our lab space. With the continued growth that I mentioned earlier, we still need all the staff, but by automating that upfront part of the process, we're able to avoid 45 FTE over the next 7 years. And the way we're doing that is by reducing the manual touches. So our lab assistants have to pour off samples into an empty tube. And so we'll take that down by 70 to 80%. The technologists that are running a lot of assays, currently our ELISA assays are quite a bit of hands-on work. So they put the samples into a liquid handler, but they're still running it to a plate washer, dispensing volumes by hand. So this will help quite a bit. And in the future state, they'll just have to add the correct reagents onto the deck. And then our long-term storage, we are currently saving previous positives. Like I said, we have to make our own controls and reagents. And those samples are already going to be in the matrix tubes that we store them for long-term. So that helps quite a bit. And then another added benefit to using those matrix tubes, we're hoping to be able to reduce the amount of patient volume that we have to request from our patients. So our laboratory uses evaluations to test. So one patient sample comes in and they can have anywhere from 1 to 24 tests ordered on that one tube. So if you think about that 5 ml tube and the amount of dead volume you need at the bottom of it, it's about 350 microliters of sample. And then we also take into consideration any repeat tests. So we need to make sure that there's enough volume for that. But the matrix tubes, the dead volume in that is limited down to 50 microliters. So hopefully we'll be able to provide a more comprehensive level of testing for patients with a smaller volume. Oops, wants to play the video again.
I want to thank you all for attending, but also thanks again to our group of speakers. Thank you for sharing your story and how the partnership with Biosero has helped to enable you to bring your automated scientific vision to life. We're going to take a moment to open it up for questions that you may have for me or any of our 4 speakers. And if you do have a question, please feel free to come to the mic so that everyone can hear that, and we'll open it up.
So this is for Merck. You have these systems that you're doing POCs, and now people use them, so now you can't develop on them. So how are you dealing with that?
That's a good question. So it does come down to, so we actually haven't gotten there yet. So it was a problem when we were doing program level support for peptide stability and we were working on the biochemical POCs. We got really careful with naming conventions, but I think the future of that is separating building different work cells in Green Button Go and trying to encapsulate the variables and stuff between them. Because you are right, if you're making change, like if you make changes to certain pieces when you're doing development work, sometimes it can affect the production level run. So yeah, we've just been lucky based off of the naming conventions, but that's something we're gonna have to look into. It's a good question.
What about the hardware side of it? Are you planning to, once you get to a certain level of usage, roll that out into a separate system? Or
Yes. Yeah, so peptide stability, for example, which is the first one we've done program support, we've built smaller versions of that to deploy to other sites. And the smaller versions just have the technology on it for that particular assay as opposed to the broad endpoint.
Okay, thank you.
Yep.
I have a question for you.
Sure.
About the 24/7 accessible hub and IT support. So is that the sales product or kind of support for customer?
Yeah, great question. So that is our new Biosero portal, which essentially is a modern distribution system that provides both how you would get your new products. It's a way of distributing the software to you, making access to the drivers available, on-demand training, so you can go in and access that training. You can access a support ticket, but it also gives you the ability to manage your automation fleet. And that's what we found in the implementation of systems is that there's no one dashboard for managing your different labs, your different instances of software, licensing, how many sockets do you have on each license, when is the last time a license was used? Is it still under support? And many things that we're seeing now is the implementation of automation is going beyond a single campus or even department, is that we're actually globalizing that. And so the team that's actually supporting it may be supporting automation across a variety of different sites. They now have real-time access into managing that, managing the users who would be able to access that capability, and they can support their team around the world through the Biosero portal. So it is both a new offering in terms of new product and continuous product support, but it also is the way that you would be able to get support tickets, training, and also in the future, it sets the foundation also for the future of having the marketplace and capability of even adding applications.
So in Japan, customers are supported by Rhodes Life Science, but in the future, so you mean we Japanese customer directly connect to Biosero, or how about that?
Yeah, great question. So we're working with Rosa Life Science now to actually roll out the Biosero portal in such a way that they would actually be able to support you in Japan, so that you have more real-time access should you need that to actually them supporting you. But it will still be through the Biosero portal. They just have an administrator capability.
So we can choose both ways.
Yes.
Thank you.
You're welcome. Good question. Any other questions for any of the speakers?
I actually have a question for the Mayo Clinic. Are all those systems GXP? Are they regulated? Like you're testing patient samples?
Yeah.
Okay. And why are they all, they're all covered in enclosures? What was the rationale behind that? Looks? Okay. Cool.
And staff safety.
Okay.
Well, thank you very much for coming. I hope you'll enjoy the rest of the SLAS 2025 event. And once again, thank you to Susie, Bill, Julia, and Ichiji. Thank you very much.
Yes, you did it.
Thank you very much. Before everyone takes off, can we get a picture?
Yeah, sure.
Maybe I'll take it back to
The hall has better lighting for all of us, so look better.
the hall does? Outside.